Hi, I'm Damon Smith, Extension Field Crops Pathologist at the University of Wisconsin-Madison. Today's webinar is going to be about bringing plant disease forecasting science to the 21st century. And what my goal here is today is to hopefully help you understand a little bit about how we develop disease prediction models, how those actually get deployed into disease forecasting systems, and then ultimately, how do we actually get those into the hands of farmers. So we're going to talk a little bit foundationally about just how we go about modeling these things and what is the science that underlies these types of tools, and then how do we actually use these tools in the real world. So let's start with a couple of definitions here, just so we're all on the same page in terms of, what really is a disease forecast. And forecasts are actually predictions about the probable occurrence or non-occurrence of a plant disease of economic importance. The preferred term that I like to use are actually disease warning systems, because a lot of times we're actually using weather that actually has already happened. We're not actually looking at a true forecast. So it'd be more appropriate to actually call it a disease warning system. Although some of the newer science, and I'll talk a little bit about how we're actually treading forward or pushing forward into the forecasting realm, I'll talk about how we're actually integrating that into true forecasts. Types of disease warnings that we can have a disease advisory, a warning that conditions were recently favorable and a disease outbreak has likely occurred. And this is what we use a lot, is actually a disease advisory type system where we're actually trying to make some fundamental decisions about how do we actually make decisions, right? Should we be putting a fungicide on or something like that? So we're actually looking at a disease advisory that essentially revolves around the disease triangle. Have we have the weather in place to actually cause a disease, and should we intervene? A forecast is a warning that conditions will be favorable. So again, more recently, we haven't delved into forecast systems because that would actually take reliable weather forecasts, although we are starting to see improved weather forecasting. So we're beginning to actually insert those into our disease warnings so that we can actually see what's coming ahead. And this would really be nice because logistically, it'd be better if we could see what was coming using our protectant fungicides to actually protect against an oncoming disease event, rather than trying to put something on that happened after an infection occurred. And from a logistical standpoint, a lot of times it's dry when we're getting these forecasts, and we could actually physically get in the field, versus if these wet events that typically drive disease actually happen and kept us out of the field. So we are striving more towards the true forecast, but we are dealing mostly typically with advisories. So the purpose of these different types of warnings and advisories are really twofold. We, probably want to make a decision about disease control. Okay, so is there even a threat? Should we even be thinking about putting a fungicide on? Has the risk been there from a weather standpoint to actually have a disease? An example here would be the white mold model, which I'll talk about a little bit later on here in soybean, where we're actually using that tool to make a decision, should we spray or not spray? Obviously, we could save some money by not putting a spray on if conditions aren't conducive, or it gives us some confidence that our decision to put a fungicide on makes some sense given the weather that's happened. We can also make disease control more efficient or improve the efficacy even of some products. Good example here, I worked in the past on a turf grass model for dollar spot. Typically, turf growers and people who deal with managing turf grass systems, say in a golf course, are always going to spray for dollar spot. But these warning systems can help really optimize the timing and make it most efficient to actually put the fungicides on, so we can get the most out of those products. So first, we can make that decision to spray or not spray, or we can improve the efficiency of our applications of fungicides. Now, what really underlies a lot of the science, if you will, that goes into these models, advisories, and forecast systems is our simple disease triangle. The triangle matters. Sometimes we use the information in this simple model, this triangle model, to make some assumptions about how do we actually apply our advisories. But What we're really doing is looking at a mathematical equation that tells us whether we've met the three vertices of the triangle. Do we have the host there? Is the pathogen present? And did we have favorable environment to cause infection and disease development? So it's really not too foundationally complicated. We're just using this simple, foundational model that underlies much of what we do in plant pathology, but we're fixing mathematical models, and we're making some assumptions to make those actually function. And I'll dig in a little bit about how we actually do that foundationally. So warning systems can be based on several different things. Okay, and I'll talk about a couple examples, and we'll start sort of on the simpler side of things, and then we'll work into some more complicated type advisories. But one of the more simple and efficient ways of sort of developing a warning system is just looking at the production or prediction of the amount of an initial inoculum. And what do I mean by initial inoculum? These are the propagules that will actually start an epidemic. Okay, so any given pathogen makes some sort of propagule, a spore, mycelial fragments, if you will, that start and initiate an epidemic. And then we have potentially with some pathogens a secondary cycle. So we'll make more spores, we make more mycelium, et cetera, et cetera. And so we can utilize that to actually look at the prediction of rate of increase. Okay, so this takes into account the efficacy, say, of a secondary inoculum. So there's a lot of sort of foundational biology work a lot of times that goes into developing some of these types of warning systems. And we can even take these two pieces of biological information and smack them together to make an even more a complicated model, if you will. So a lot of times we're starting with simple biology, knowledge that we have about a particular pathogen, how much inoculum might be there, and then using information that we might know about how efficient that secondary inoculum is to actually develop these types of warning systems. Just to give you an example of how this has been functioning actually for quite some time, a lot of you may be familiar with Navitude testing programs, and these are really a form of a disease advisory, and these have been going on for quite some time. The 1950s and 60s, we had nematode advisory programs in Europe and the US. C.J. Nussbaum and K.R. Barker at North Carolina State developed soybean cyst nematode testing programs and started, deploying those in North Carolina, and those have since spread to much of the soybean production region of the US. Again, you probably didn't think about this as being a disease advisory, but it really is. We're taking nematode counts, which is our initial inoculum, okay, and we're affixing some warning based on some information that we know about the soil types and those sorts of things. Just to remind you just a bit about the disease cycle for soybean cyst nematode. Of course, the nematode scientific name, heteroglycines. Overwinters, remember, is those eggs which are contained inside these cysts. And these cysts will break open and spill those eggs out into the soil. We get juveniles hatching. The juveniles migrate to a soybean root tip, invade that root tip, start feeding. The females give rise to more eggs, and then the cycle continues. So we have a measure of initial inoculum. We also know a little bit about secondary increase. We know that this does have some secondary increase in season, but that secondary increase can be rather slow relative to, say, foliar disease of corn. So again, you probably didn't think about this as an actual disease advisory, but that's essentially how we're looking at it as scientists and trying to deploy a warning about what to do, what decisions should we be making. Now, obviously, we're not going to be making decisions about controlling nematodes in season, but we can take this information and we can use it to make some decisions about what varieties should we be planting, and should we even be planting soybeans in a particular field. This is just a table that shows kind of a sort of a simpler decision guide that helps us sort of work out what kind of risks do we actually have. So you can see the risk indice over here on the left hand side, and then we have the egg counts in per 100 cc's here. And notice that this is broke out between soil types. So we know that sandier soils give us a little higher risk. Therefore, you can see our risk levels here in terms of eggs per 100 cc's is lower in a sand than, say, in a silt or clay. And you probably know this if you work in soybeans or do any type of soybean cyst nematode sampling, you'll know that we sort of adjust things. So again, this is an interesting type of advisory. We've been doing this a long time. Notice there's no real weather parameters built in here. We'll be working towards that here in a few slides. But we're taking that information that we know about initial inoculum, secondary spread, soil types, and some of those things to build out an advisory that can then guide decisions for the next season. Now, increasing in complication are, models where we actually look at, not only where is the initial inoculum coming from, how much is there, how fast is that secondary cycle, but we're, also starting to overlay some weather information on top of that, especially wind trajectory information. So we can look at, this is especially useful when we look at pathogens that don't overwinter in a particular region. A good example of this is actually the soybean rust epidemics that we see every year in the southern U.S. Typically not a problem in the far northern or upper Midwest, and there's a good reason for that. The spores just can't overwinter, and they don't like the environment in that upper Midwest region. However, we can affix some advisories based on some things that we know. If we know where soybean rust is present at any given time, we can then use wind trajectory models to actually look at or forecast when we might actually see a disease develop and where it might actually develop. And so again, a little more complicated. We're starting to build in some of that weather information, but we're still using underlying biological information that we know about. Where can these spores overwinter? What plants are they overwintering on? And then where is that inoculum actually spreading to? And we're using simple, scouting to find those point sources. Once we know where the point sources are, we can then, we use that wind trajectory modeling to actually see where the next risk might be based on those wind currents. So again, this is a little more complicated model, still looking at initial inoculum and also secondary spread, but we're starting to build in weather information to improve our accuracy of the modeling prediction. Now, this brings us to a conversation in terms of environment. So as we start to think about that disease triangle, I've been really focused, on the pathogen and host sides of those triangles initially here. And we haven't talked much about the environment except for this wind trajectory example and soybean rust. But we do know that for many pathogens, we also, are looking at temperature and moisture variables. So these things typically favor pathogen infection, pathogen development, especially as we are talking about fungal organisms most of the time here. And there's different types of measurements of these variables, and we might use even combinations of environmental variables to sort of build these out. So we know typical moisture variables, relative humidity, leaf wetness, and leaf wetness duration is a very useful one from a pathology perspective, although it can be hard to directly measure that. Rainfall can be very important, but highly variable from one location to the next. And then for our soil-borne pathogens, soil moisture is a very important variable as well. But modeling soil moisture can be difficult. Some of you who deal with soils might know this, but trying to get accurate soil moisture data without actually directly measuring it is really difficult, although the science is improving there. Temperature, both air and soil temperatures, are also very common variables. They're also fairly easy to measure and get a hold of that data. So you'll see these commonly used in various modeling exercises. Now, we use a lot of underlying biology, information, and science to build these out. We're not just pulling weather parameters, out of thin air and trying to fix these to certain systems and then hoping that these things will work. We do a lot of work trying to understand what parameters actually play a role. And I'm going to use this white mold cycle here in soybeans just as an example here, how we actually build one of these models out, what kind of science goes into this. We take a lot into consideration as we develop these types of tools out. We're not just applying mathematical formulas here, again, and hoping that they work out. So I'll just remind you of the soybean white mold cycle here. So we start here with these survival structures. We call these sclerotia. These are those hardened, melanized little structures that resemble rat turds or rat droppings, and they're usually found in the upper 2-inch profile, soil profile. These give rise to a very important structure here, which we call an apothecium, and this is basically an inverted mushroom-like structure. The reason why this is so important is this is what leads to spores and then also spore dissemination into open soybean flowers. So in soybeans, the white mold fungus, Sclerotinia sclerotiorum, its primary infection happens through these blooms. So between the R1 growth stage and about early R4 growth stage is the window of opportunity for the fungus. So right there, you know that, just from a foundational standpoint, that's probably where we really need to focus in terms of starting to develop a warning system or some sort of forecasting system for this disease. As the fungus infects, it goes down into the stem, and then several weeks later, you start to notice that bleached part of the stem, and eventually white mold starts to grow, and we get the formation of more sclerotia. Once we run our combines through the field, those sclerotia then are dropped off those plants back onto the soil, and we just deposited more inoculum back to the soil. Now, we've looked at this in detail in my laboratory. We worked with others, Michigan State, also Iowa State University, trying to understand what weather information really drives the development of this mushroom. We know that this mushroom has to be present to give rise to the spores. And without the spores, we don't get infection. So we took a sort of a deep dive look at what really drives the biological development of this little mushroom structure. And I'll just jump to the chase right now that we know that these are some really important parameters here. We need cooler temperatures, especially daytime high temperatures need to be on the lower side. So those cooler days, we have to have high humidity that coincides there. And we have to have canopy closure of 40% or more between rows. So we have to have dense canopies in order for these things to actually develop. Okay, so the formation of these little apothecia, here's an image of those over on the right-hand side of your screen. You can see, we can have multiple apothecia on a single sclerotium, actually, depending on the size there. We know that we have to have these things present in the field, and they have to give rise to those spores to infect the plants, okay? Now, I want to just convince you of this so that I can convince you that modeling the mushroom development is worthwhile, okay? And so we did a series of experiments, and I just want to give you a little bit of science here just so you can understand where we're headed and how these models actually function and where we're getting these risk indices from. What we did is we set out gridded plots, and these were replicated plots. So each of these little boxes right here is a 10-foot wide plot of soybeans that's 20 feet long, that we have a 5-foot alley. So we have these grids across the field. We go in, lay transects of these cool little purple plates. And these purple plates tell us, they do a little color change from purple to yellow when the Sclerotinia sclerotiorum spores land on that plate surface. So we can actually enumerate how many spores are actually flying. And we lay these plates across a transect of these grids. And then we also place some plates around the corners of the field. Okay, so what we're doing is we're sending undergrads out on a daily basis. They're going in, they're actually looking for the little mushrooms. While they're looking for those mushrooms, we lay these trap plates out in a transect across the plots, and then we're using those plates to try to trap spores. So we're taking 2 bits of information there, the mushroom formation along with spore flights. Now, let's look at some numbers here. So here are some of these grids, okay? And then we have, there's actually 2 different fields here. So we have 2 different grids. We have days after seeding here. So again, we're dealing with soybeans here. And then we have the growth stage in parentheses of those soybeans. So really interestingly, the development of apothecia starts to occur right before blooms happen. Okay, so the more intense the red inside these boxes, the more mushrooms we actually have inside that 10 by 20 plot. Okay, so as that canopy's filling in close to R1 and we start to get blooms, you see that we're starting to ramp up in terms of the number of mushrooms actually forming. This is all biology, and this is, well-timed out there. It's not a happenstance that these apothecia are forming right at the flowering time. They sort of know, if you will, that this is how they have to infect. So you can see that we get a sort of a peak in terms of apothecial development right around that full bloom timing. And then as we approach the onset of potting, you'll see that the numbers of apothecia start to decline. So we have a peak of formation, and then that declines back off as we get later in the season. Okay, so that's the mushroom development part of this. We also have, remember, that trap count data. So here we have the average apothecia per plot. So the average number of mushrooms we have in these blue bars. We're just looking at one set of plots right here in this particular graph. And we have some growth stages affixed here. So we got the beginning of bloom, full bloom, and then the end of bloom right here. And then this orange line is the average number of ascospores caught per trap each day we trapped. And you can see there's a little bit of lag. We get the onset of these mushrooms, and then, ascospores pick up, which makes sense. We have the mushrooms form, then we get those spore flights. And then as the mushrooms go away, we get fewer spore flights. Okay. So that's really interesting. We also know, based on that previous slide, that, these apothecia tend to be very aggregated in the field. You remember they were sort of off to one side in that grid. And so we can use this information to know that from inside a soybean field, that's where we get most of our infections. They're going to be from usually a fairly localized area, and we have to have the mushrooms present in order to get the spores to infect. Okay, so that's the window in this case for the fungus. And sure enough, if we come back and we actually look at these grids again later in the season and actually see how much disease is there, you can see that disease basically starts right where we had those mushrooms form. Okay, so if we understand where they are present in the field, maybe through mapping, and then we use weather information, we can really start to target our management strategies to where disease is going to develop. The more intense the red here, the higher the disease levels. Here we are at the R6 growth stage, so we're getting close to full maturity here. And you can see a lot of infection over here where we had large numbers of apothecia. Okay. So hopefully this convinces you that, if we can figure out how to predict that mushroom formation, we can understand whether we're at risk or not for infection of the white mold fungus on soybeans. Okay. So we took all that mushroom data, if you will. We also had weather stations deployed, and we're looking at remote sense weather information and all sorts of data sources. We had 9 site years of data, and basically we started doing some mathematical modeling to relate the weather that we recorded to those apothecial counts, those mushroom counts that we had taken year after year after year in these grids. And we started to develop the model. So the weather information basically predicts whether we have presence or absence of those apothecia. So it's foundationally a fairly simple modeling approach. The real work takes place trying to understand the biology and doing all the data work to acquire the counts to actually get the apothecial data. So once we did the math work, we can start to develop these probabilities. So essentially what we're doing is we're taking that weather information and we're predicting the chance of actually seeing the mushrooms develop. Okay. This is a strip trial location, some work we did with a farmer in 2017 up in Marathon County, Wisconsin. What you're looking at here on this vertical axis is the probability that there's going to be mushrooms present. Red would be high, yellow medium, blue low, gray basically not active or just barely active. And then we have some growth stages of the crop. So again, we're just looking at conducive weather here. You can see that, from just a weather standpoint, the risk can be very high actually prior to bloom. Okay. But there's some very important things, remember, that you have to take into account on the soybean side of things. Remember, I said you have to have 40% canopy closure or more. Right? You typically don't get that till around R1. So that typically is an on-off switch for you. Especially if you look at our tools that we've developed, we actually ask you, has the between row canopy actually closed? Yes or no? And that turns these models on and off. But you can see here in this particular field, as we approach R1, we're pretty high all through bloom time, reaching maximal risk actually right as bloom's starting to end and we're getting into the pod development time, and then it dives a little bit and then expands again. We can actually look at what was going on in that field. So here are the actual aerial strips in the field, and they had some strip trials in there. And at that point, they were using the approach fungicide program there, which typically is a two-pass where you come in at the start of bloom and then again towards the end of bloom. And you can see we had some checks out here on the edges of the field. But what was really interesting when we started comparing single pass applications, we had equally good control of a single pass at R3 as we did with both the two-pass control and a three-pass program here. So what was going on? Well, you'll remember in the probability risk, we had the highest probability during bloom time right at that R3 growth stage. So again, if we're thinking about optimizing the efficacy of these products, we can use these tools to sort of maximize, the best time to actually apply that. And you can see much improved over the non-treated or even a single application or product at R1. So, we did a little bit of good work here by putting that R1 application on, but not quite the work that we did with a well-timed R3 application. So again, these tools can help you make a decision on whether to spray or not, or can help you optimize the application timing of these products. Just to look at some data from the most recent field season that we went through, I went ahead and included some efficacy trial data from Hancock, Wisconsin in 2022. We had various standard programs. This is a trial we do every year where we insert various programs from different companies. We run them head to head, and we just test efficacy and then take yield data. So a little bit of a busy slide here. Let me set this up for you. So on the vertical axis here, we have disease index score, which is percent disease essentially. And then we also have yield here. So in the blue are the disease index scores that we took at R6, so near full maturity. And then the orange here is the yield in bushels per acre. And then you can see the various programs here along the bottom axis. And you'll notice here we have several treatments in here that we call Sporecaster. So this is the tool that we've developed. We're using the models that I've just talked about in the previous few slides here and developed this tool that we call Sporecaster. So our mathematical model, it pulls down weather information, runs it through, develops a risk index, and then tells us when we should be spraying. So we simply turned our brains off, if you will, and applied our applications based on this Sporecaster tool. And you can see where the two cases where we had Sporecaster, we really maximized the control. And I will say that these were applied around the same time as the R3 applications. So these were fairly late timings. Makes sense. That's when we had good canopy closure, when the weather was most conducive. And you can see we really maximize yield here in terms of bushels per acre. You'll also notice that, combining our Sporecaster tool also with application technology. So we had 360 undercover nozzles here. We were able to further improve our efficacy of these particular products, when paired with that Sporecaster tool. We did have a couple of check treatments in here. So our two-pass R1, R3 Endura at 8 ounces per acre. This is usually a standard. We wouldn't recommend this program because it would be quite expensive to apply. But we use it to look at the best case scenario. And you can see where we combined our Sporecaster technology also with, our 360 undercover nozzle technology. We had really excellent control and really good yields compared to some other programs in here. So again, just an example of where we've been headed, how well these tools have been working, and we've had good luck with Sporecaster over the years with really, recommending the optimal time of application for our fungicide products. Let's just switch gears a little bit. We'll switch crops and diseases just to give you another example about, how are we actually utilizing science and biology about various pathosystems to develop these efficacy type tools or these forecasting tools. And tar spots been a disease on everybody's mind lately in corn. It's been a couple of big epidemics in recent years, and we've been using our modeling tools to try to develop some new models again, weather-based type tools to, try to optimize applications and trying to, really get an idea when should we be actually applying our fungicides. Just to show you the disease cycle, we know that the fungus, the tar spot fungus, can overwinter in the upper Midwest on corn residue. The ascospores then are ejected up into the corn canopy. Typically, we see disease develop in the bottom part of the canopy and then work its way up, and that typically happens when it's rainy, kind of overcast, and humid. Temperature is important, but not as important as the moisture components here, and I'll explain that a little more here in a bit. And then different from white mold, where we have really just one cycle per season and one sort of time where the fungus can really infect, which is during that bloom time. That's different than the tar spot fungus, which can have repeated cycles. So once we get some infections happening, we get more spores that will infect more leaves, so on and so forth. And so we start to see an exponential increase as we go through the season. Once the plant is covered by tar spots, eventually it usually succumbs to disease, dies, and dries down, and then we go in and harvest and complete the cycle. Now, why are diseases like tar spot emerging here in the upper Midwest? Well, it's really tied to climate and weather. So weather comes back to haunt us again here with this particular example. Just to show you, some past trends here. These are the 30-year precipitation trends: June, July, August, and September. These are 2020 slides here, but it shows a good example of how our trends have been towards wetter climate. Okay. The other thing that's happening too in our systems is we have shorter rotation. So we're typically more on a corn or corn soybean rotation. We have no-till cropping systems where we leave a lot of residue there. And that's all combined again with a, on average wetter season that we're having, or wetter than, the 30-year average at least. And we all know that August, in recent years has been actually some of our wetter months in some areas of the upper Midwest. Could be drier in some areas too, but, for the most part, where we see these tar spot epidemics, has been trending on the wetter side of things. And just to drill in a little bit about what has been known about the biology of this particular fungus based on, weather and how weather influences the tar spot fungus, there was some work done in the mid-90s looking at these. Temperature, of course, important. Cooler overall average temperatures typically were reported in that particular paper, although we're starting to learn that, this fungus might slow down a little bit when it gets warmer, but it doesn't really, those higher temperatures aren't lethal. And so the fungus can survive. Once the cooler temperatures come around, we have some wetness, it can take right back off. What you will see here, though, is that 4 of the 5 parameters that we show here in bullets, those are moisture parameters. So relative humidity greater than 75%. If we have a lot of leaf wetness at night, foggy days, 10 to 20 foggy days per month, high rainfall, 6 inches or more rainfall in this particular set of studies. So moisture really important, drives a lot of what happens here, along with a little bit of temperature. Over here, this group took this weather information and extrapolated this on the upper Midwest, the corn production belt. This was published in 2018 prior to the first real big tar spot epidemic. And basically what they're showing here is that, inherently our climate in the upper Midwest is fairly conducive for this particular fungus. The deeper dark red is a probability of 70 to 100% chance that, if the inoculum's here, we're likely to see tar spot develop. And I think the last few years are pretty telling. We sit in a very conducive climate in general for this particular disease. Now, in terms of our more recent modeling and what we've been doing, we've been through a few iterations, and that's another thing I'd really like to, hammer home here, is that once we develop a model, we typically don't just leave it alone. We take more data, we retrain those models, and we revalidate. So we're not just building a model, we're leaving it, and we never, let it grow, if you will. We're taking new data and we're putting that in. So we've been through several versions actually of the tar spot modeling, and we're actually on to version 3.0 this year. We're actually going to be developing version 4.0 pretty soon. So again, it's an iterative process, and our latest modeling again really points to the moisture components here. We do have temperature data in here, and then we do take into account whether a fungicide has been applied or not. That really dictates that secondary spread, and that tells us how fast is the fungus actually recovering. But if we're actually looking at weather parameters, it turns out that a 14-day range of weather typically has been pretty informative for us here in the Midwest. So we're looking at mean temperature over a 14-day period. We're looking at the 14-day minimum relative humidities. We're looking at 14-day mean dew points, and then we're also looking at the 14-day total rainfall. We build all this into our tool, and then we, on a daily basis, we can redo the predictions and what the risk is that we're actually going to see tar spot develop. So what you're looking at here is how do we actually take that understanding of the disease triangle and actually affix it to our tar spot or risk probabilities. So the whole foundation of our tool is pretty similar to what we were doing with the white mold models, except now we're looking at actual tar spot development. So that's the data we took. Then we're pulling that weather information down from the cloud, and we're putting that model together in much the same way. So we're looking at 40% risk. So anything above 40% is high. So anytime you see a blue bar, that means that the risk for that particular day was calculated to be high. And sort of how you understand or interpret this is it's similar to a weather forecast. So today might be a 70% chance of rain, if you will, okay, or 100% chance of tar spot development. That's how you sort of look at this. And how you prep for that is, well, if it was a rain forecast, if it was 70% chance of rain for today, you'd bring your umbrella with you, okay? And so we're doing the same thing here. If we're at 40 to 100% chance or tar spot, we're probably going to be making some decisions on intervening, putting a fungicide on. Now, what we did differently in this modeling is that we actually looked at spore flights through the field as well. So these green bars show you 7-day windows where we actually had spore flights detected of the pathogen. Okay, so now we're overlaying the inoculum. Okay, so we know that the fungus was present in these green windows. We can also take into account the growth stage of the corn crop during that time, and then look at the weather information and whether we had a high risk. Did we have blue bars? So we can start to actually hone in on these discrete times through the season where we have that disease triangle met. Where did we have pathogen? Where is the weather conducive? We had a couple of different types of spore traps out there, so we can get a little different type of data as well in terms of when that was actually occurring. And then we can actually take and start to overlay our disease development and growth stage models on top of this. So this is a real deep dive, layered approach into that disease triangle, trying to understand when is that triangle met and when are we going to start to actually see disease develop. And this red line is basically displaying when that disease is really starting to take off. But the key here is that, we had a window of conducive weather in here. We also had pathogen showing up on several different cases here, depending on which spore trap type we were using. And you can see the onset of that disease coinciding roughly with when we had spore flights and conducive weather conditions at the right growth stages in corn. Now, fundamentally, we call this the incubation periods and latent periods. So the incubation period is the time from when we see basically spore flight to disease development, and then the latent period is the time from spore flight to where we start to see secondary increase. And those are really important times when we start to consider our fungicide protection intervals. And that's why, we're looking at this, and we get a lot of questions about, how long does it take from when a spore lands on a leaf to when we actually see disease show up? And we have data here to dig in and really start to answer those questions so that we can further guide even our fungicide application intervals. So we're also expanding the science to really dig in and help fine tune those management techniques. Just to show you some data again from the field on how well, tools like this could work. We run again a set of rodeo trials similar to what we do with With soybeans, this is data from 2021, so the tar spot epidemic that was pretty significant here in Wisconsin, at least in 2021. And we did use tar spotter along with our Veltima 7 fluid ounce rate Veltima fungicide treatment here. So tar spotter actually told us to apply a 2-pass program. So we actually had our first application at V10, followed by a second application at R2. And you can see that that was one of the best programs here. What you're looking at with this line right here, these are what we call disease progress curves. So we take several ratings through the season, and you can kind of see how it's increasing, and we get into this exponential increase where the line gets pretty steep as we rate. And then we can take this information and we can boil it down into a single number here. So the bar chart is the same data as the line graph. It's just a little easier on the eyes. And you can see that some of the lowest disease levels or lowest disease intensities were from the tar spotter applied fungicides. So again, if we choose the right product, we can really optimize the application timings of those products, and it was better than anything else from a disease standpoint in there. Now we did have some other programs applied right around that tassel R1 timing that was on par. So anything inside the red box here was not different from each other, but you can see a lot of these things much better than the non-treated check. So again, really optimizing those fungicide programs and getting those applications on at just the right time. Just to show you a nice visual, here we are scouting with our camera, just to compare, visual health of the corn crop. So on your left, there's the non-treated fungicide. On the right is the fungicide trials where we had tar spotter, predict those 2 applications. We're looking at the R5 growth stage here. This was September 7th in those plots. You can see there's tar spot in that right-hand image there when we zoom over a leaf, but you can see how much better that crop looks over the non-treated control. And that was pretty consistent where we ran the tar spotter tool. Now, again, it's not perfect. Like the white mold models and any models, none of them are perfect. They're models. They get us closer to what's really happening, but they are informative and can help really guide those decisions. Now that we have a foundational understanding of, kind of how these models work, I just want to take a few minutes just to have a conversation about how do we actually get these in the hands of farmers and practitioners? How do we, bring that science actually out to the field so that we can make it useful for actually making these decisions, so that you don't have to call up, myself or, any one of my colleagues in each of their respective states and trying to get, prediction on whether you should be spraying or not. And what we found in our program is that, folks really like their iPhones. And so we went that route, utilizing the smartphone. So we're using apps here to take that information and, bring it out to the real world. Now, in making that decision, we went through, a thought process like, what are the things that make these warning systems useful? Why would we actually implement one? And so here's just some rules that we sort of follow in guiding us and how we actually deploy these tools. We want to make sure that they're reliable. They're based on data. I spent a lot of time here trying to convince you that we're using science. Our models are underlying with quite a bit of data and work, and we continue to refine those things. We're also looking at diseases that are important but can be sporadic. The environment's not always going to be conducive. We may not always have inoculum in an area. Again, thinking about that disease triangle, we want to make them as simple as possible, and we try to develop the simplest models. Sometimes that isn't always the most accurate, so we do have to adopt more complex models. But if we can embed them in a smartphone app, we feel like we can take those complex models and make them more user friendly. We also want to make sure we have effective disease management tools available. So we do have more tools now in terms of chemicals and equipment and application strategies and those sorts of things. So we're keeping that in mind. We want to make sure we have appropriate assays and monitoring systems so we can understand that biology again. And then we want to make sure things are cost effective. And so we take all of this into consideration when we're actually trying to figure out how to actually get these in the hands of users. there were some great publications in the early 2000s just talking about how do we actually increase adoption, because at that time, we didn't have smartphones, and this was a big conversation, because there are other disease forecasting tools out there. I've just talked about a couple of examples that we've worked on, but how do we actually get those out? How do we, make them usable? And at that time, a lot of the user interfaces were online. We used online dashboards. Forecasts were long decisions a lot of times at that time. Somebody would have to go out, look at their weather station, bring the weather data back in, run it through a spreadsheet, et cetera, et cetera. So one of the rules that we borrowed actually from this McGarry et al. Paper is the cup of coffee rule. So in that paper, they talk about, a farmer to really think about how to get a farmer to adopt a forecasting system. They need to be able to run that forecast and get their decision in the time it takes them to drink a cup of coffee. And so again, the smartphone platform really fits this well. We can do all the math on the phone. We have the GPS information there. We can tell the phone to go grab weather information somewhere, and then do all the math in seconds on that phone. So we're talking about, being able to go from not knowing what to do to getting a forecast and making a decision in as little time as, say, 30 seconds or so. So that's been really important in helping us sort of think through this process. And so all of this is basically culminated into a couple of apps that, are freely available. These are research-based tools. Again, the models that I've talked about today underlie each of these tools here. You can access these. These are free tools. They were supported by the Wisconsin Soybean Marketing Board in the case of the white mold tool. And then we also have the Wisconsin Corn Promotion Board and also the National Corn Growers Association when it comes to the tar spotter tool. So those are out there for you to download. Those are the QR codes there. Or you can go to the Nutrient and Pest Management Program website, and the URL is right here at the bottom of the page. So feel free to check these out, load them on your phone, work through these. You can run forecasts on a daily basis anywhere. It'll grab the GPS on the phone and actually find the weather information. We use cloud-based weather on a 4-kilometer grid to develop these risk indices, and you can start to make some better informed decisions about how this should actually work. And we know that there's some barriers to this type of work, but we feel like, again, it's keeping some of the things in mind in terms of how do we actually move these out. We know that one of the barriers is farmer age. The average age of the farmer is 57 or higher, although we do know that there are new farmers getting into the business, and that average age is much younger, more along the lines of my age, in their early 40s. So I've heard that argument before, that not everybody has a smartphone. I know the internet is not accessible in a lot of rural locations, but these are efficient ways of trying to at least deploy helpful tools and get them in the hands of people. The other thing that can be a barrier is the quality of the weather. I've talked a lot about weather and how we used it in modeling, but I didn't really talk much about how we're actually utilizing the weather. And when we first started using cloud-based weather, we were using an API called Dark Sky. And basically, Dark Sky is a weather aggregator. And then they interpolate the weather between set grid points. So they're utilizing lots of different weather models. So this was a list at the time of different types of models that they pulled into their aggregation, and then could fix those to different size grids. And so there are some drawbacks from the weather accuracy standpoint, although we're finding that is rapidly improving. And so we monitor this on a yearly basis. We put hardwired weather stations out, and we're looking at weather quality on an annual basis, and we do change and revalidate and fix things as we catch errors. The other thing is we know that the grids can be rather wide. It's hard to get a within field forecast. So usually these are more at the field level. We do strive to get smaller grids, but there are some hurdles there again in terms of accuracy and how we actually measure that. So again, age and then quality of weather information are two big hurdles, although we are working through these. And we do believe we have tools that are, in the realm of about 80 to 85% accurate. So That's really pretty good and much better than a coin flip, which is how we used to make our decisions. The last thing I'll just leave you with here is, in addition to the freely available tools, we also have tools that were developed using our models through a company called Field Profit. Field Profit adds some unique bells and whistles actually to our tools, in that mainly they can give us an actual true forecast. And I mentioned at the beginning, that where we'd really like to be in the future is actually having true forecasts. So what you're seeing here in this video is we're setting up a field in our tool here. So this is very similar to what we do even in the freely available apps, where it hits a location, and then you can also grab a map and find a point here, a GPS coordinate point. So we're running a tar spot tool here in this view, and we're going down to Florida because it was in the middle of winter when we developed this. So we set our GPS point, and then we're going to hit run here, and we get an instantaneous forecast by just giving a couple of pieces of information about when the last fungicide was applied, what's the growth stage of the crop, do we have irrigation, and now we're actually going to go in and get our forecasts. So rerun the models, and you can see the 7-day look ahead is actually quite conducive. Another nice thing about Field Profit is it gives these trend lines. One thing we've learned over the years with both the white mold models and the tar spot models is that if we look at the 7-day trends, that's usually more informative than just an instantaneous look at what what the weather looked like for that particular day. So I would encourage you to look at the trends over time, whether you're using the Field Profit version or the Sporecaster or Tar Spotter versions, and then investigate this Field Profit version, because that forecasting tool, especially the 3 to 4 day forecasts, we found can be quite accurate there. So again, just a QR code if you're interested in downloading that tool. Again, the underlying formulas and everything are the same between Sporecaster, Tar Spotter, and Field Profit. So with that, I'd just like to thank my lab. They've done quite a bit of work on all of this. You can see them there in the upper left-hand corner. We have lots of funding agencies, both in Wisconsin and externally, that help support a lot of the foundational biology and science work here. And then also entities that helped us develop the apps and tools there. So I hope you found this webinar informative. I hope you have a better understanding about how disease forecasting tools work, and really the science that underlies these very important tools that are now deployed and widely available to growers and practitioners around the country.